Hello, my name is Jacqueline Jingri, and I work for CAGEF, which stands for the Center for the Analysis of Genome Evolution and Function. We are physically located in the Department of Cell and Systems Biology at the St. George campus of the University of Toronto. I'm here to talk to you today about our proteomics platforms and their applications and some of the cool things that they can do for you. Our proteomics platforms are a liquid chromatography system coupled to a mass spectrometer for LC-MS analysis and a Via-core surface plasmon resonance system. Our mass spectrometer is a thermoscientific LTQ-XL mass spectrometer, which is your high sensitivity, high resolution, and high mass accuracy instrument. It is coupled to an exigent liquid chromatography system. Our chromatography system is a nanoflow system, allowing for easy coupling to our mass spec and offers superior sensitivity compared to a conventional flow system. Our SPR system is a Via-core 3000 instrument from GE Healthcare. This instrument is a powerful probe for biomolecular interactions at a surface for both qualitative and quantitative studies. Now I'm going to talk to you about our mass spectrometry platform. What can we do for you with this platform? What if you have a mixture of proteins from a co-IP, or co-immunoprecipitation? That's where you precipitate your protein or proteins of interest out of solution by using an antibody that binds specifically to your protein. Or what if you have proteins isolated from tandem affinity purification, that is your TAP-TAC protein? Or what if you have a protein in a gel band and you would like to identify that protein or proteins in your sample? Or you want to know whether it has post-translational modifications, or is it upregulated or downregulated? These are questions that can be answered with LC-MS analysis. Now I'm going to give you detailed examples of some of the things we can do with the mass spec platform through case studies. In the first case study I present to you, we were interested in whether protein X autoacylates. That is, put a cell group on itself. So the intention was to search for acetylation sites on protein X. In this example, protein X was isolated through co-minute precipitation. The protein sample was subjected to reduction, alkylation, and trypsin digestion. Trypsin was used in this case, but other enzymes can also be used. Following overnight digestion with trypsin, peptide enrichment was done, and the sample was injected onto the LC-MS column, onto the liquid chromatography column, where the peptides underwent separation under gradient dilution. Gradient dilution is where you basically gradually increase the percentage of organic solvent as separation proceeds along the column. The effluent from the column was then electrosprayed into the mass spectrometer for MS analysis and detection. So the peptides are separated by liquid chromatography and detected with the mass spectrometer. The mass-to-charge ratio for peptides in the sample is detected and recorded as an MS1 spectrum. The peptides are then fragmented using collision-induced dissociation, and the fragment ions are detected. The fragment ion or MS spectrum can then be used to identify a peptide. In this study, the peptides from the MS data were searched for acetylation using the global proteome machine search engine and searched against our in-house database since the protein of interest is not present in publicly available databases. The output from the search engine was viewed and validated using Scaffold proteome software, which outputs a list of top protein hits with a protein identification probability and number of unique peptides. It also shows any post-translational modifications specified in the search and outputs a lot of other cool ways of visualizing the results. So your results will be given to you in the form of a scaffold file. For your own exploration, web links to the GPM search engine and the scaffold building software are provided. So in this case study, we were interested in finding acetylation sites on protein X. LC-MS analysis was done on protein X and its catalytic mutant for negative control. We searched for acetylation sites on serine, threonine, and lysine residues, and the output from the search engine validated scaffold. What you see here are screenshots from scaffold with the results of our experiment for protein X and for its catalytic mutant form. The first thing I want to highlight is that sequence coverage information is given. We got a 51% sequence coverage for protein X and 59% for its catalytic mutant. Highlighted in green are the modifications found in the protein. In this case, we found one autoacetylation site on a serine residue in protein X. This was not seen in its catalytic mutant. So that was good news for us. We found evidence of autoacetylation in protein X. Further, if we zoom in on the right panel in the screenshots taken for scaffold for both samples, we see a list of all peptides detected. For each peptide, you can see charge, mass, position in the peptide sequence, confidence scores, and modifications. Here for protein X, we see acetylated peptides, but none in the catalytic mutant. Here are MS or fragment ion mass spectra for the acetylated peptide found in protein X and its unacetylated form found in the catalytic mutant. As I said before, there was no acetylated form of this peptide detected in catalytic mutant samples. We found one key site. The first MS spectra represents an acetylated peptide from protein X with the acetylation site. The second MS spectra represents the same peptide, but it's from the catalytic mutant form of protein X. Here, no acetylation is seen. The molecular weight shift for the intact ion in protein X versus its catalytic mutant is 42, corresponding to an acetyl group. If you look at the most intense peak in purple in the mass spectrum, these purple peaks that you see here. It corresponds to the doubly charged dehydrated form of the molecular ion. The acetylated form is seen in protein X and the unacetylated form in the catalytic mutant of protein X. And once again, the mass shift is 42, corresponding to an acetyl group. I've also shown a singly charged Y ion, or protonated ion, as another example. This is shown in red. The acetylated form is seen in protein X, whereas the unacetylated form is seen in the catalytic mutant form in protein X. the mass shift here, once again, is 42.1 to be exact. So we can conclude that protein X autoacetylates. The second case study is a continuation of the autoacetylation work. Here we're interested in deducing whether protein X acetylates a suspected target. LC-MS analysis was done on 3 samples. The target protein in the presence of protein X, and the target protein in the presence of the catalytic mutant form of protein X. The first mass spectrum is the fragment ion spectrum of target protein alone. The second mass spectrum is the fragment ion spectrum of the acetylated peptide in the presence of protein X. And the third spectrum on the right, it's the MS-MS spectrum of the unacetylated form of that peptide found in protein X, found in the target protein in the presence of protein X. In the presence of protein X, we found one acetylation site on its suspected target. In the suspected target alone, no acetylation sites were found. In the sample with the target plus the catalytic mutant form of protein X, no acetylation sites were found, but in the target protein, in the presence of protein X, one acetylation site was found. So we can conclude that protein X acetylates its suspected target. So these are 2 case studies on mass spectrometry work done at CAGEF. The first being the detection of autoacetylation in protein X. The second being showing that protein X acetylates its suspected target. So now I'm going to talk to you about our second platform, our BA-Core surface Plasmon resonance instrument. So what can we do with this platform? What if you have a complex mixture of possible interactors and you want to pull out specific interactions with a particular protein? Or you have 2 purified proteins and want to know whether they bind to each other, or how strong is the interaction, that is, measure KB, or how fast the association is, that is, measure your on and off rate constants. Or you want to measure the energetics of the reaction, determine enthalpy and entropy. Well, our BA-Core surface Plasmon resonance system will be able to answer these questions for you. Before I present to you a case study highlighting some of the work done on this instrument, it's necessary for me to explain how the instrument works. The basic configuration, or Kretschmann configuration, of most commercial SPR instruments comprises the following components. You have a light source, a detector, and a prism onto which a sensor chip is mounted. The way the system works is that light passes through a prism and reflects off the backside of the sensor chip surface to the detector. At a certain angle of incidence, known as the resonance angle, light is absorbed by electrons at the surface of the sensor chip, causing them to resonate. These resonating electrons are known as surface plasmons and are sensitive to their surrounding environment. The consequence is an intensity loss in the reflected beam and is seen as a dark band and can further be seen as a dip in the SPR reflection intensity curve. The shape and location of the dip is a sensitive probe for interactions at the surface of the sensor chip. So how is this technique used as a probe for biomolecular interactions at the surface? Usually, one molecule is immobilized to the surface of the sensor chip. In Biocor terminology, this is the ligand. A solution with its binding partner or the analyte is flowed over the surface of the immobilized ligand. Binding of the analyte to the immobilized ligand is reflected as a shift in the dark band and a shift in the SPR dip originally seen. This dip shift can be used to monitor real-time binding events at the surface of the sensor chip. In our third case study that I present, we were given two GST purified proteins and asked to answer the questions, do they bind to each other, and is it a specific interaction? So, in our experiment, protein A was immobilized onto the surface of a sensor chip. And this allowed us to establish our baseline. Protein B was injected over the surface of the sensor chip, and this allowed us to monitor binding of protein B to protein A. After some time, buffer was injected over the surface, and dissociation of protein B was monitored. The surface was then regenerated for another set of binding dissociation experiments. The on-rate constant can be extracted from the association phase, while the off-rate constant can be extracted from the dissociation phase. The ratio of these rate constants gives the binding constant. I should mention that there are a variety of sensor chips available for use in these types of experiments, but what is used would depend on your molecule of interest and what molecule you're going to be able to mobilize on the surface. This would have to be discussed with us before you submit your sample. So in the third case study, we were interested in answering the question, do the proteins bind to each other, and is it a specific interaction? So protein A was immobilized onto the surface of the sensor chip, and protein B was flowed over immobilized protein A, and binding was monitored. What you see on the left here are dose response curves for binding of protein B to protein A. So a series of binding experiments were done with a concentration range of 1.5 to 500 nanomolars of protein B. As the concentration of protein B increases, there is an enhancement in the SPR response, indicating binding of protein B to protein A. Another set of similar experiments is done for binding of a negative control to protein A. The results are shown here on the right. There is no enhancement of the SPR signal at any concentration of the negative control. So the negative control does not bind to protein A. We therefore conclude that protein B binds specifically to protein A. In the fourth and final case study I present to you, we're trying to answer the question, what is the strength of the interaction between a purified protein X and its binding partner, peptide X? Protein X was immobilized onto the surface of the sensor chip. Dose response curves were generated for a peptide concentration ranging from 1.5 to 500 nanomolars. The experimental dose response curves are shown in color. These curves were fitted to a one-to-one theoretical binding model, and these are the curves shown in black. The output reported was the on-rate constant, the off-rate constant, and the binding constant, as well as a chi-square value to assess the goodness of fit. It's not shown here, but we were able to reproduce this data several times. And so we were able to quantify the strength of the interaction between two molecules. I should mention that the software has other models, other theoretical models, that is, which can be used to fit experimental data, such as the 2-to-1 model or an equilibrium model. Here is the overall proteomics workflow for both of our platforms. For the Spec platform, or the LCMS platform, samples can be submitted in the digested or undigested form. If it's undigested, we would perform the digestion. Following this, LCMS meta-analyses will be done, and a database search and a scaffold report will be generated for you. For the BIA-Core platform, if sample is submitted to us, the first thing we would do is scout for optimal experimental conditions. Optimal conditions being what molecule to immobilize on the surface, selecting the right sensor chip, selecting the right method of immobilization, buffers used, how to identify specific interactions, designing control experiments, how to fix for non-specific binding, and the regeneration of the chip for use again. The data will then be evaluated, fitted to an appropriate model, and a report generated for you. Here is a summary of the different types of analyses that can be done on both our platforms. For the LCMS platform, we can do proteomic profiling. We can ID proteins in complex mixtures, from co-IPs, from tap proteins, proteins in gel bands. We can also identify post-translational modifications for you. For the BIA-Core platform, we can measure on and off rate constants, which will allow us to get a dissociation constant. We can also measure the energetics for biomolecular interactions. And we can do this for protein-protein interaction, protein-peptide interaction, protein-small molecule interaction, drug-target interactions, and receptor-ligand interactions. And of course, if there's any other sort of interactions that you're interested in, you can talk to us and we can decide whether it's feasible. Here is the contact information for our center. Your general inquiries can be sent to CAGEF@utoronto.ca. Our director is Dr. David Gutman, our facility manager, Dr. Pauline Wang, and our project manager, Dr. Silva Donaldson. We would like to thank the Ontario Genomics Institute for organizing and facilitating this webinar. We would also like to thank the Canadian Foundation for Innovation and the Ministry of Research and Innovation for research funding, and of course, you, the audience, for participating in this webinar. Thank you, and I will now address any questions that you may have.